So I will talk about my postdoc project with the name PartiCell, and it's about understanding this very long title that was mentioned. To give a little bit of background, it is known that the inhalation of particles can result in lung inflammation and chronic lung diseases, such as chronic obstructive pulmonary disorder, fibrosis, or even lung cancer. And there have been some epidemiological studies that indicated evidence of adverse effects in the respiratory system of workers exposed to such engineered nanomaterials. However, the mechanisms of lung inflammation and also fibrosis, especially the particle-cell interactions, are not quite yet understood. And in the light of the ongoing process to reduce animal or in vivo studies, there is a need of advanced physiologically relevant in vitro models to study pulmonary exposure of nanoparticles. However, there is a lack of these models, so my study hoped to contribute to this, filling the gap. So in our study, we were interested in titanium dioxide nanoparticles, as these are one of the most widely produced and used nanoparticles worldwide. And they are also important for the Norwegian industry. As you can see a picture there of Kronos Titan, which is located in Fredrikstad, Norway. Quite a big titanium dioxide manufacturing plant. And titanium dioxide has various applications, ranging from food and drug colorant to inks and paints, cosmetics, and as you might know, it is found in sunscreens to block the ultraviolet radiation. And it also may be used in batteries or in drug delivery applications and so on. So the aim of our project was to increase the understanding of the cellular events that are involved in the occupational-related development of chronic lung disorders due to this titanium dioxide nanoparticle exposure. It has to be noted that titanium dioxide was classified as possibly carcinogenic by IARC in 2005, I guess. As chronic inflammation is a key characteristic of carcinogenicity, this might be an important endpoint to study. So specifically in our project, we wanted to develop and optimize an advanced physiologically relevant cell co-culture model representing the alveolar barrier in the lungs. And using this model, we tried to assess then the time-dependent immunological responses following TiO2 nanoparticle exposure. So how did we set up our model? Well, we used cell culture inserts with a semi-permeable membrane. And we had 3 cell types, one representing epithelial cell type on top of the insert, and also macrophage-like cells, which are immune cell type. And on the bottom of the insert, we cultured endothelial cells. And this kind of represents the alveolar barrier. So we have the air-blood barrier. This also means that the cells on top are exposed to air, while the cells at the bottom, they are surrounded by medium. So this was our cell culture. And the particles we used were rutile and mixture titanium dioxide particles. And they were dispersed in a kind of artificial lung surfactant, just to stabilize them and have them in the nano-sized form. And the exposure, for the exposure, we used this VitroCell Cloud 12 system, where you have these wells, and then you can put the cell culture insert into these wells. And you put a chamber on top of the instrument, which is connected to a nebulizer. on top there. And on this nebulizer, you add the particle suspension. And applying a current to this nebulizer, it starts creating this cloud or liquid aerosol inside the chamber. And this cloud is then evenly kind of deposited and distributed to the wells and to your cells. So we had various post-exposure time points ranging from 4 hours to 14 days. So we were actually able to keep our cells alive for 14 days, which is pretty good for such a complicated cell culture. And after all these time points, we looked at cell viability, gene expression and release of inflammatory markers, and also biodegradation of the particles in the cells. But I will not go into detail with that today. I will show you some results from the characterization of the particles. You can see some pictures of rutile and the mixture titanium dioxide particles. And these images were taken after the particles were dispersed in this artificial lung surfactant. The primary particle size was about 20 to 30 nanometers, and after dispersing them in this lung surfactant, this size increased to about 100 to 150 nanometers. But it was kind of a small size distribution of the particles, so that was the aim in our study. So here you can see some of the results from the exposure. I haven't mentioned that we used 2 different doses, 1 microgram and 5 microgram per square centimeter, and these were calculated based on the occupational exposure limits for ultrafine titanium, and also based on the kind of limit from the instrument. And here you can see heat maps from showing the upregulation of different genes. And what is interesting to look at is this interferon gamma, which is circled in, and this is upregulated for all the particles, for all the doses, and basically at all time points. So this was kind of the most interesting finding. And we can also see that we have an upregulation of this DNA damage marker, GUT45A, for P25 after 14 days, so after kind of long-term exposure. And we also see a bit of upregulation of a second inflammation-related marker, only for the P25 particles. But it has to be noted that at the same time, we did not see any decrease in cell viability. So that was very important. So it's kind of a subtle change in inflammation-related markers. So with this, I'm ready to summarize some of the findings. So we thought that this in vitro ALI model represents a valuable tool to assess the cellular effects of nanoparticles up to 14 days. And inflammation seems to be kind of a major mediator of this cell damage in our alveolar barrier model system exposed to titanium dioxide nanoparticles. And these findings are also supported by the literature on in vivo studies using mice and rats. However, further studies are needed to optimize this model even further and to study the exact mechanisms behind. So, and with this, I'd like to thank the occupational toxicology group at STAMI and our kind of international partners in the US at NIOSH and also Luxembourg. So thank you for your attention.